Very good afternoon. I'm Aisha and I will be your host for today's webinar. Kindly note that this webinar will be recorded. It's my pleasure to welcome you to our ACTRIS Cell Therapy Lecture Series. Today's lecture by our distinguished speaker, Associate Prof. Lin Ye Jin, focuses on development and clinical applications of virus-specific T cells. Prof. Lin graduated from NUS in 1987 and obtained her MRCP in 1991. She has been working in the Department of Hematology since then and is actively involved in clinical service, research, and teaching. Her areas of interest are in hematopoietic stem cell transplantation and cellular immunotherapy. She has published various transplant-related areas, cytokine-induced killer cells, and virus-specific T cells. She is the principal investigator of clinical studies on usage of CIK cells for leukemia post-autologous and allogeneic hematopoietic stem cell transplantation with fundings from NMRC and SingHealth research funds. Before we get started, if you have any questions during the presentation, please type them into the Q&A window, and Prof. Lin will address these questions after her presentation. And now, without further ado, let's welcome our distinguished speaker, Prof. Lin, please.
Okay, thank you, Aisha. Can you hear me?
Yes, I can hear you well, and I can see your screen too. Thank you.
Okay, thank you. So good afternoon, everyone. First, I would like to thank ACTRIS for this invitation to share with you today on this topic of virus-specific T cells. So this is the outline of my talk. For the first half, I will talk on the background of VST for treatment of EBV-related malignancies as cell therapy for drug refractory virus infection in immunocompromised hosts and off-the-shelf VST. Then I will share our preclinical work in the preparation of setting up of a third-party VST bank. Firstly, let's look at VST for treating EBV-related malignancies. So EBV is an oncovirus that is implicated for Hodgkin's disease, non-Hodgkin's lymphoma, NK lymphoma, post-transplant lymphoproliferative disease, which PTLD in short, and also nasopharyngeal carcinoma, NPC in short. After an acute infection, the EBV genes will downregulate so that the surface antigen expression will be reduced to evade immune surveillance. So there are three types of latency represented that represents the immunogenicity of the EBV-driven malignancies. So in type 3 latency, there's an expression of a wide array of EBV proteins which are highly immunogenic, and such tumors happen in immunocompromised hosts such as after solid organ or marrow transplant called post-transplant lymphoproliferative disease. Type 2 latency has less antigen expression and includes in lymphoma as well as NPC. And type 1 latency is immunologically silent. So we'll talk about EBV T cell against type 3 and type 2 latency tumors. So immunogenicity of PTLD makes it susceptible to cell therapy. And EBV T cells has been explored since more than almost 3 decades ago. This is the earliest study in 1996 from the Baylor's group on the use of donor-derived EBV T cells in marrow transplant, which is given as a weekly infusion, and it demonstrates that the infused EBV T cell can expand in these patients and can mediate EBV-specific cytotoxicity, and they can persist for up to 18 months and can further expand upon rechallenge with the antigen. This is from the same Baylor group years later in 2010 with long-term follow-up of patients, a large number of post-allo transplant patients given donor-derived EBV T cells. For prevention of PTLD, none of the 101 patients developed PTLD, which is to be compared with historical control where there were 5 out of 42 that developed PTLD. For treatment of PTLD, 11 of 13 patients achieved a CR without recurrence. GVHD risk is low, and the authors commented that in comparison, rituximab, which is the standard first-line drug used in treatment of PTLD is suppressive to B cell function and does not confer long-term protection. So what about lymphoma with type 2 latency? This is not in transplant setting, therefore it uses autologous EBV T cells. EBV T cells are generated by autologous dendritic cells transduced with adenovirus vector that express LMP2. And in vitro testing shows good cytotoxicity against autologous lymphoblastoid cell line or PHA blasts that express LMP2. These are the two blue lines up here. Clinical outcome, patients with Hodgkin's or non-Hodgkin's lymphoma at high risk of relapse treated with these EBV T cells, 28 out of 29 did not relapse at a median of 3 years. And for relapse or refractory disease, 13 of 21 patients responded to cell therapy. And besides LMP2 specific T cells, other antigen-specific T cells could also be detected, and this is a phenomenon known as epitope spreading. This is from a Korean group for patients with NKT lymphoma, which is a very aggressive disease with poor prognosis. So EBV T cells were given to 10 patients who have completed all the standard treatment, which includes an autologous stem cell transplant. And with further treatment with EBV T cells, the 4-year progression survival is 90%, which is a lot higher than what is expected. So this table shows the cell dose and the lack of toxicity and the survival of these 10 patients. So in summary, these earlier studies testify to the potency of EBV T cells in EBV-related lymphoma. Now, what about another latency 2 tumor induced by EBV, which is NPC? Okay, so this is pioneering work from the Baylor group, where they use autologous EBV T cells for advanced NPC, which showed modest efficacy like 2 CR in 6 patients with refractory disease. It shows feasibility and safety. The same EBV T cell was also studied by our oncologist colleagues at the National Cancer Center for metastatic or locally recurrent NPC. In this phase 2 study, patients received 4 cycles of chemotherapy and then followed by 6 infusions of EBV CTL. This is from the clinicaltrial.gov website on the phase 3 trial involving 330 patients who are randomized to chemo alone without or with EBV T cells. Unfortunately, there was no clinically significant difference in terms of survival outcome, but this is the online version and we await the final manuscript. If there are oncologists in the audience with the insight into this study, I will appreciate very much your sharing of your thoughts later. So now let's move on to VST for virus infection in immunocompromised hosts. So while EBV T cell was studied for EBV-related malignancies, another application that was explored for virus-specific T cells is in the treatment of opportunistic virus infection in immunocompromised hosts. So to date, the most widely studied patient population is that after allogenic stem cell transplant. Patients after allo transplant or solid organs transplant, or patients with primary or acquired immunodeficiency states because of their very profound T cells impairment in T cell immunity, they are susceptible to opportunistic virus infection. The virus that are most often implicated are CMV, BK virus, EBV virus, adenovirus, and HHV6. These viruses can cause a whole range of serious disease and death, such as EBV and adenovirus that can cause multi-organ disease. CMV and adenovirus that can cause multi-organ disease. EBV, as we mentioned, can cause PTLD. BK virus can cause hemorrhagic cystitis and BK nephropathy, and HHV6 can cause encephalitis. So antiviral drugs are used for some of these conditions, but it is toxic to the marrow and toxic to the kidneys. And some patients develop resistance to the drugs. And for BKV, there is no good effective treatment. So just to illustrate the severity of the problem in a post-allo transplant population. So in this analysis of 404 patients after allo transplant, this bar chart shows the rate of infection due to each of these viruses in the first 3 months. You can see that CMV and BK virus represented by the green and the purple bar charts are highly prevalent. So a patient can have more than one virus reactivation after transplant, and non-relapse mortality will increase with the increase in the number of virus infection. And so patients, for example, with 4 virus infection at any time have a much higher mortality rate and a lower survival rate in the first year. So CMV disease is the most problematic virus infection after allo transplant, especially this three decades ago. So there was no effective treatment for CMV. So this 1995 study is the earliest to generate CMV clones from from donors using a tedious method. The CMV T cells was generated by repeated stimulation with CMV virus and with feeder cells and with cytokines, and the clones were selected by limiting dilution assay. So CMV T cells were given to 14 post-transplant patients weekly for 4 infusions, and they showed that the transferred clones could expand after repeated infusion and can persist for more than 12 weeks, and none of the patients developed CMV disease, viremia or disease. And because virus often coexist in post-allo transplant immunocompromised patients, the ideal product will be one that are reactive against multiple viruses generated from the same culture. So this is one that shows the culture of trivirus specific T cells that is possible against CMV, EBV, and adenovirus. So at this juncture, we will digress a bit to elaborate on the method of generation of VST. This review is summarized very nicely the evolution of method, which has been simplified over time. So the earlier clinical studies used EBV transformed B lymphoblastoid cell lines as antigen presenting cells to grow EBV T cells. To add on additional antigen, for example, for trivirus specific T cells, then the autologous dendritic cell or the EBV lymphoblastoid cell lines are transduced with adenovirus that also express CMV PP55 antigen to produce 3 virus antigen stimulation. These methods are tedious and it takes like 3 months to generate the virus specific T cells. So subsequent improvement involve using DNA plasmid transferred to the dendritic cells by electroporation. And now the most commonly used method is actually the commercial overlapping pool peptide, which consists of about 100 or so 15 mers with 11 amino acid overlap spanning the whole virus antigen. And simply by loading onto the mononuclear cell and then ex vivo expanded with an optimized cytokine cocktail. Besides ex vivo expansion, to obtain VST, the other method is actually direct selection by magnetic column of VST, either by multimer binding of the virus peptide MHC complex to T cells, or capture of interferon gamma secreting T cells after pool peptide stimulation. So this is a method to select VST by the multimer technology. So the principle is that T cells will recognize specific peptide MHC complex on the target cell through their specific T cell receptors. So peptide MHC complex that binds these T cell receptors can be made, and then they can be tetramerized for better binding to the TCR, and the T cells can be isolated this way. So the VST obtained by this way is pure, but the disadvantage is that it is based on MHC restriction, and commercial multimers are only available for the common HLA alleles, which therefore is not applicable for everyone. And also there is a potential for viral evasion because only one specific viral epitope is targeted. This is another isolation method making use of interferon gamma secretion to isolate the VST. So cells are incubated overnight with pooled viral peptides, and those T cells that recognize the viral antigen will secrete interferon gamma, and the interferon gamma can be captured labeled by interferon gamma capture reagent and incubated with anti-interferon gamma microbeads, and then it can be selected using magnetic column. So this will bypass any limitation of HLA restriction and will select polyclonal T cells that recognize multiple viral epitopes. However, the purity is low and they may be contaminated by alloreactive T cells, and which in turn will limit the number of VST that can be infused because of concern of GvHD. This is from a UK group using this gamma interferon capture method to isolate CMV T cells from marrow donors. As you can see, the infused T cell dose is low in the range of 10,000 per kilo, but there will be in vivo expansion. The median purity is not high at about 40%. They reported protection against reinfection in the majority of patients. In fact, HSA and NUH labs have the capability to do this procedure. Now back to our multivirus specific T cells after I have introduced those methods. So this study uses multivirus specific T cells generated by stimulation with pooled peptides, as elaborated earlier, of antigens from these 5 viruses, and the effectors are from the original stem cell donor. This small study shows efficacy as all the patients with active viral infection responded. And in these plots, you see that the dotted lines which shows the viral load, there is a drop in the viral load, while the solid line shows the CMV T cells as measured by ELISpot assay expand over time. It shows safety as there is no GvHD or systemic inflammatory response. Of note, this is the first time where BKV and HHV6 T cells are used to treat these infections. But understandably, the spectrum of antiviral reactivity is limited by the donor's immune status against that specific virus. That means donor has to be immune to the virus in order to be able to produce the virus-specific T cells. So that brings us to the next development of VST, which is from third-party donors, frozen in a bank for use as an off-the-shelf product, just like drugs. So far, what I have shown are studies using allogenic donor-derived VST for virus infection after allo transplant. But the problem of donor-derived VST is that they are not available in cord blood transplant, and in some unrelated donor transplant, you may not get further donor cells. And as stated earlier, it is also not useful if the donor is immunologically naive to the virus. So one other very important problem is the lag time to the availability of the cell product for therapy. Even using direct selection, which is an overnight procedure, it takes time to screen the donor, book for leukapheresis slots, and lab processing slots are all these logistic issues. So in opportunistic virus infection, patients who fail drug therapy are often in need of alternative therapy very urgently. Therefore, third-party off-the-shelf VST becomes the goal that is hotly pursued nowadays. The earliest study came from this Scottish Blood Bank, which is published in 2002, which was a bank of EBV T cells derived from healthy donors for the treatment of EBV PTLD in post-transplant patients. So in 8 patients with progressive PTLD, 5 patients completed the treatment, of whom 3 had complete remission, and it is mainly in those whose tumor are early or are localized and polyclonal. So they rightly also stated that the frozen bank is less prohibitively labor intensive and expensive for wide-scale use. The same group updated in 2007. This is a multi-center study involving 33 patients from 19 centers with EBV PTLD after all sorts of transplant who failed to respond to standard conventional therapy for PTLD. The dose used was not high at all, like 2 million per kilo, and they gave weekly dose, giving a 64% response at overall response at 5 weeks and 52% response at 6 months. They observed that closer HLA matching correlates with better outcome. This is for post-transplant opportunistic virus infection, and using a third-party bank of 32 tri-virus specific cell lines. It uses the method of generation was a genetically modified EBB LCL presenting all the 3 virus antigen. So 18 cell lines was given to 50 patients and between 1 to 3 infusions with mostly 1 to 3 HLA allele matches, and they reported overall response of 74% at 6 weeks. And they also reported very low incidence of GVHD. They further expanded to target 5 viruses, and similarly for post-allo transplant patients. So these VSTs are generated by stimulation with the pool peptide method that I elaborated earlier. So that was a bank with a size of 59 VST lines. 38 patients will receive these third-party T cells. The response rates at 6 weeks is high at 92%, and some patients required retreatment. Persistence was short-lived, but these are only partial HLA match because these are only partial HLA match, but this is not the critical problem because it only serves as a bridge until the patient recover their own immunity, and then it is not required anymore. And there is no problem of severe GVHD. So this slide summarizes the main studies on third-party bank, either for clinical trial or clinical service. Again, as you see here, these are the centers: Baylor, Memorial, Sloan Kettering, the Scottish Blood Bank, Sydney, and Cincinnati. So it's again single or multiple viruses with various methods of generation, and the dose is actually not high. Dosing is often repeated weekly. And the patients are mostly after allo transplant and supplying up to 100 patients in some studies. The clinical indication is drug failure or proven drug-resistant mutations, except for BKV, where there is no good drugs and BKV T cell is used as a first line. Safety data is very reassuring. As you see, GVHD risk is low, and outcome is strikingly consistent in that response rate is in the range of 70%. In fact, off-the-shelf VST is on the way to commercialization. So this is by biotech company collaborating with the Baylor College to produce VST for various viruses and at different stages of clinical trial. And that sets the stage for a commercial off-the-shelf VST product. They published their study on posoleucel, which is again 6 viruses, including JC virus, okay, for post-allo transplant patients with one or more of these viruses. The starting cells come from healthy donors, stimulated by pool peptide, similar to their previous studies in culture method and clinical protocol. They reported treatment of 58 patients and giving an overall response rate at 6 weeks of 95%. Of the 23 patients who had refractory BKV cystitis, the resolution of hematuria at 6 weeks was 74%. And they showed that 59 VST lines could provide for 97% of the requests and was rapidly available within 48 hours, which is an important fact for such third-party T cells. So matching was at between 1 to 5 of the 8 HLA alleles, and median is at 2, matching at 2 alleles. There's no significant clinical difference in the response and the degree of HLA matching. There was a reduction in viral load over 2, 4 to 6 weeks, while the frequency of the VST in the peripheral blood against CMV, BKV, adenovirus increases after infusion. So after reviewing the current status of VST, let me share with you our preclinical work on VST. First, we asked the question, is there a demand for VST in our patients? So with the help of my transplant colleagues in SGH and NUH, This is the outcome of a survey on the incidence of reactivation of 5 viruses after allotransplant. The most prevalent virus is, of course, CMV, which is about 37%, and of whom about more than half were so-called problematic, in that causing disease or required prolonged hospital stay or had recurrent cycles of infection, or patients developed toxicity or even contributed towards death. The prevalence is lower for adenovirus and BK virus. For EBV, there is also a significant number of EBV reactivation requiring treatment with rituximab, and of whom about half were more serious, resulting in PTLD. So for BKV, a fraction of patients with BKV cystitis in the range of 10%, and it can be very challenging because there's really no good treatment. BKV is also a problem for renal transplant patients. This is information from renal transplant colleagues, where about 1/4 of patients get BKV reactivation, and 1/3 of them can be persistent, and a handful of them will develop proven BK nephropathy in the renal graft. So we can say that while it's not an issue for the general population, in this immunocompromised host, VST has a role to play when drug treatment is ineffective. So I can say that there is an unmet need. Okay, so with this background, I now present our work in the generation of VST. We have grown VST against CMV using our CIK method, and VST against other viruses using method published by the Baylor's group. And I will elaborate on our preparatory work for establishment of a third-party VST bank, including consideration of the bank size. First, a very brief background on cytokine-induced killer cells. So CIK are polyclonal T cells that are expanded under cytokine combination stimulation, and comprise mainly of CD8 subset with a smaller proportion of CD4. And there's a subset of CD3 cells that upregulate CD56 that has the highest cytotoxicity. Target recognition is non-MHC restricted, and it has shown anti-leukemic activities for minimal residual disease states after allotransplant, and there are also many clinical studies on autologous CIK in various solid tumors. So by the mechanism of target recognition, CIK is not antigen specific. So we explored integration of method of generating CMV T cells into CIK platform by providing CMV pool peptide stimulation to generate CMV specific CIK. So ficod lymphocytes are incubated with CMV pool peptides, and these are those that are CMV reactive will upregulate CD137. And after 16 hours, anti-CD137 is added, and these cells are selected by magnetic column. So this is a crucial step because CD137 or 41BB serves not only as a marker for selecting the CMV reactive T cells, but also as a co-stimulatory signal that induces the proliferation, production of cytokines, increased signaling, and amplifies the T cell cytotoxicity. So the selected cells are cultured under CIK condition, and we do functional assay at maturity. So with this method, CMV CIK that are specific against CMV targets can be generated. These are seven separate killing assays. The red lines show killing of autologous PHA blasts loaded with CMV peptide as CMV targets, while the blue lines are against control PHA blasts without peptide. Cytotoxicity at 1:1 is between 14 to as high as 51%, and at 10:1 is above 50%. So since the purpose is for off-the-shelf use, we verified that frozen CMV CIK can still kill target cells upon thawing, although at about 10 to 15% lower than the fresh counterparts. And after culture, CMV CIK are mainly CD8 T cells as compared to pre-culture lymphocytes. And compared to starting lymphocytes, they have less naive T cells, they have less early effector T cells, and instead there's a higher proportion of late effector T cells. And actually, this may explain the clinical observation of a lower incidence of GvHD when polyclonal CIK is used in an allogenic setting. CMV-CIK are oligoclonal in contrast to polyclonal starting lymphocytes. So the CMV-CIK are shown in as red bars here, and the blue bars represent the starting lymphocytes. There's an uneven distribution of some TCR-V beta families in contrast to the even distribution of the pre-culture T cell. This is an extreme example with the V beta 3 family expanded to constitute 75% of the cells in the culture as compared to a much lower percentage in the starting lymphocytes. We published our findings a few years ago. And CMV-CIK is actually directly translatable for clinical use because we have previously manufactured CIK for clinical trial. These pictures show the maintenance, the harvesting, washing, and cryopreservation of CIK in the GMP lab at the cell therapy facility at HSA. So we next tried to generate CIK with specificity against other viruses using the same method. Okay. But despite many attempts and many modifications, this method failed to generate CIK against other viruses. The possibility is that the CTL precursor frequency against viruses, except for CMV, is low in the starting lymphocytes, and therefore providing co-stimulatory signal to 4-1BB could not preferentially expand this minute population. And under CIK condition with OKT3 and IL-2, the non-specific T cells are expanded instead. So we then adopted the published method by the Baylor Center for Cell and Gene Therapy with some modification. So white cells from discarded clones from platelet-pheresis donors were taken to isolate mononuclear cells and stimulated with pool peptides, and then expanded under a different set of cytokine, which is IL-4 and IL-7, for 2 to 3 weeks in G-Rex flask. So this picture shows the culture in G-Rex flask and the cell clusters under microscopy. We monitor the expansion for T cell subsets and did cytotoxicity against autologous and allogenic PHA blasts loaded with the respective virus peptides. And after optimizing the method as a preliminary step towards a third-party VST bank, we started to screen healthy donors using their discarded cone cells to characterize the potential of each donor in generating VST. So for those donors with good killing, we did HLA allelic level typing. And we built a database of 49 donors whom we have screened and characterized for their antiviral activity, with the eventual objective of using clones from these selected donors to generate VST lines for a third-party bank. To generate, these donors has to have potent cytotoxicity and broad-spectrum cytotoxicity. This chart gives a sense of the frequency of CTL precursor against each virus in healthy people, as measured by the percentage of CD137 upregulation after 16 hours of incubation with the respective poop virus peptide. So for example, this flow plot shows that in the bulk culture, 1.5% of CD8 cells and 0.58% of CD4 T cells upregulate CD137 in response to CMV poop peptide stimulation. This chart shows that in the 38 samples that we assayed, about 60 to 70% of donors had CTL precursor of more than 0.1% against the 5 viruses. And the CTL precursor frequency is the highest against CMV, while the rest are much lower, but they are comparable against amongst each other. We did cytotoxicity assay of single and the 5-in-1 VST against autologous PHA blasts that are loaded with the respective adenovirus, VKV, CMV, EBV, and HHV6 pool peptide. You can see there is a broad range of cytotoxicity amongst donors in their killing potential against the various viruses. So CMV specific T cell has the highest cytotoxicity, median level of like 59%, which is significantly superior over cytotoxicity of VST against other viruses. If we take 30% as significant cytotoxicity, this cytotoxicity above 30% was seen in 50%, 42%, 56%, 56%, 42% of a single virus, single VST against adenovirus, BKV, CMV, EBV, and HHV6. And for the 5-in-1 VSTs, as shown in the pink columns, you can see the cytotoxicity is also comparable to the single VST in their specific cytotoxicity against each of these 5 viruses. This table shows the T cell phenotype, T cell subset of the VST, which consists mainly of CD4, more CD4 than CD8 T cells, and with minimal NK cells or NK-like T cells. So ask the question, what is the spectrum of cytotoxicity of this 5-in-1 multi-VST? So amongst the 36 multi-VST lines, cytotoxicity was present in 16 lines against adenovirus, 9 lines against BKV, 20 lines against CMV, 14 lines against EBV, 12 lines against HHV6. So you get the idea that not every line is reactive against all the viruses, every virus. The next question is, how broadly do these multi-VST lines cover? Do they cover all viruses? So amongst the 36 multi-VST lines, 3 VST lines were cytotoxic against 4 viruses, 8 lines against 3 viruses, 13 lines against 2 viruses, 9 lines against 1 virus, and 3 lines, there are 3 lines that did not kill any virus. So we show that from healthy donors, we can generate VST lines, either single virus specific or multi-virus specific, and quite a proportion of them, but not everyone, have good cytotoxicity. But the ultimate aim is for third-party use rather than autologous use. So it is more relevant to ask whether these effectors can kill and how well they can kill allogenic targets. This is a tabulation of the cytotoxicity of CMV T cells against autologous and allogenic targets presenting CMV peptides. These are effector target pairs, and the HLA alleles that are restricted for CMV peptides and shared between the effector and the target are in red. This column N is the total number of such CMV restricted HLA alleles that are shared between the effector and the targets. So in this top panel, you can see that effector against allogenic target showed very good cytotoxicity, which is comparable to autologous cytotoxicity. And all these pairs had 2 or 3 shared alleles that are known to be CMV restricted. In contrast, in the bottom panel, there was none or only 1 shared CMV restricted alleles between the effector and the target, and they showed negligible allogenic cytotoxicity despite significant autologous cytotoxicity. So this gives an important proof that sharing of at least 2 virus-restricted alleles between the VST line and the potential recipient can result in good antiviral toxicity, which is only logical because viral peptides are presented in the context of self-NHC to their cognate T cell receptors. So besides potent cytotoxicity for third-party use, another relevant question is how big the bank has to be in order to provide cell lines for as many patients as possible, and yet cannot be too big for cost consideration. To answer these questions, we have to first know the HLA diversity of our local population. This table on HLA allele frequency of Singapore Chinese is extracted from a paper from Dr. Ren Ichi's lab. It lists the HLA with allele frequency of more than 5%. And let's call these locally prevalent HLA alleles. So you can see it's a narrow spectrum where the HLA alleles with frequency more than 5% is limited to only 6 HLA-A, 6 HLA-B, 5 HLA-C, and 7 HLA-DRB1 alleles. You can easily understand that the diversity of the HLA composition of a population is relevant in deciding the size of a third-party bank. So the next question is, can these so-called high prevalent, locally prevalent HLA alleles present CMV peptides? This is an online immune epitope database which gives a comprehensive list of HLA alleles that has been described as restricted for CMV. Those alleles that are locally prevalent with allele frequency of more than 5% are highlighted in green. So it is really nice that many of the local prevalent HLA alleles have been reported to be CMV restricted alleles. And so this implies that the chance of generating VST line with one or more of these HLA alleles can present CMV to an unrelated recipient is high, simply because many of these alleles are highly prevalent in our local population. To answer the question of how big must a third-party VST bank be, we did a simulation for CMV T cells from our database. So we typed HLA class 1 and class 2 alleles for 24 of the VST lines that has good CMV cytotoxicity as listed in this table. In the second last column, the score reflects the number of high prevalence alleles that each CMV line has. So in fact, as you see, it is quite high. The score is quite high, and 18 of the 24 CMV VST line has at least 5 or more high prevalent alleles. The last column here shows the number of CMV restricted alleles rather than just shared HLA alleles. So CMV restricted alleles that each VST line has. And again, 17 of these 24 VST lines have at least 5 or more high prevalent alleles. So this shows that our database of CMV T cell lines, a high proportion has high prevalence HLA and are also CMV restricted HLA. So this is due to the fairly homogeneous HLA composition in our local Chinese population. And so from this list of 24 VST lines, we chose 6 lines highlighted in green here that are good CMV killers to form a hypothetical cell bank to test how well can this small bank of just 6 CMV T cell lines cater for our patients. We obtained HLA typing of 100 patients who have undergone allogenic transplant, so they are readily available information. The ethnic distribution of these 100 patients is representative of our local population. We then see how well can our 6 cell lines, how well can these 6 cell lines be matched with the HLA A, B, C, DRB1 for each of these 100 patients. So this is done with a simple Excel formula. The number of shared alleles between each patient and each of the CMV line was computed. So for example, for patient 1, he matches no HLA allele match with cell line 9, 1 allele match with cell line 16, 17, 21, 4 allele matches with cell line 27, 2 allele matches with cell line 28. Okay. And with that, the number of CMV T cell lines that matches 1, 2, 3, 4, 5, 6, 7, 8 alleles with this patient can be computed. Okay. So this was done for each of the 100 patients as shown here. Okay, so with that, we came up with this table that summarized the matching between the 6 CMV T cell lines and the 100 patients who serve as hypothetical recipient to simulate a scenario where we need to find a cell line for a patient from this bank of 6 CMV lines. So the way to read this table, for example, there are 10 patients who can find 2 VST lines that matches at 3 alleles. Similarly, 16 patients can find 1 VST line that match at 3 alleles, and 2 patients can find 3 VST lines that match at 3 alleles for a total of 28 patients who can find CMV T cell line who are matched at 3 alleles within this bank of 6 VST lines. So in total, 99 of 100 patients, as shown here, okay, can find CMV cell line that match at least 1 allele. 92 patients can find CMV lines that match at least 2 alleles, and 79 patients can find CMV lines that match at least 3 alleles. This is a similar table, but we analyze for more stringent criteria of matching for CMV restricted HLA alleles instead of matching just for matched HLA alleles. Okay. So remember that previously we showed that many of the locally highly prevalent HLA alleles are also CMV restricted alleles. Therefore, it's not surprising that there are also many patients who can find CMV lines matched at 2 or 3 CMV restricted alleles, such as shown here. So this simulation of an actual scenario demonstrates that at least for CMV, a bank size of just 6 CMV cell lines can provide for vast majority of local patients with VST line that match at least 2 alleles. And these third-party VST line will have reasonable chance of killing CMV infected tissues in these allogenic recipients. We publish our results demonstrating that a small number of well-characterized donor can provide VST line to cover for our patient population, and that this is a cost-effective strategy. So with those work, we are now ready to embark on the project of setting up of a third-party VST bank. Firstly, we upscaled the production from G-Rex 2 cm square base area to G-Rex 100 cm square. We will then transfer technology to GMP facility, and they will need to do 3 validation runs. After that, we will generate T cell line from these well-characterized selected donors when they come for their next platelet pheresis, and hope that they will agree to give their clones again. The plan is to offer for compassionate use under the salvage innovation therapy under MOH CTGTP directives, collect enough clinical data, and make my 3rd attempt to apply for NMRC grant to do a proper clinical trial. With that, I finished the talk. I have many people to thank, including my research officers, collaborators at blood bank and at cell therapy facility at HSA, and of course platelet donors, and without them this work would not be possible. Thank you for your attention, and I welcome your questions and comments.
Thank you, Prof. Lin, for a very elaborate presentation. So we have one question from our audience, from Prasad. Prasad says, thank you, Prof. Lin, for a fantastic talk. If I understand correctly, GVHD risk in third-party VST is a function of dose of T cells, type of infused T cells, and the degree of mismatch. Are the cells caught by IFN gamma capture all RO memory cells? What are the guardrails in terms of doses and mismatch in the third-party VST used in allogenic BMT setting without causing problems and antigen presentation?
Okay, thank you for this question. Obviously, someone with a lot of insight into this. So for the gamma interferon capture assay, usually we get donors. If the donor is full match, we are not so worried, because for full match donor, we can give donor lymphocyte infusion up to at least 1 million per kilo CD3 cell per kilo, and without much concern. But a lot of time we cannot get from the matched donor, but rather we have to get from some family donor that hopefully are partially matched. So that is how usually interferon gamma capture system we make use of this gamma interferon capture system to give, for example, CMV T cells for patients using their half-matched family donors who has shown to have reactivity against this virus. So you are right, for haplo donors, there is a much lower threshold of the non-virus specific T cell, meaning the non-interferon gamma secreting T cells, the limit is like 5 × 10⁴ per kilo, as we would give for a haplo matched T cell as a first donor lymphocyte infusion. Yes, so for that reason, it may limit the contamination of alloreactive T cell, may limit the number of virus-specific T cells that you can give for these patients. But even so, a small number, we have done a few, like 2 or 3 patients, even a small number of a few thousand CMV-specific T cells can expand quite remarkably in vivo. Yes, does it answer your question?
Thank you, Prof. Lin. We have another question from Jai Chandran. What is the level of HLA matching required for off-the-shelf VSTs? Is there any correlation between level of HLA matching and incidences of GVHD or allo rejection?
Yeah. So all those published studies all did HLA matching at the allele level, meaning the 4-digit HLA. And most but not all the studies shows that better matching correlates with better response, which intuitively should be the case. In our in vitro study, we also showed that at least 2 allele matching of the restrictive the virus restricted HLA is important for effective cytotoxicity. So in terms of GVHD, so these are immunocompromised hosts that is worry of GVHD, but I believe it is because these are expanded, so for a third-party bank, right? These are all cultured expanded T cells. They are mainly late memory T cells rather than naive T cells. So their capacity for causing GVHD is much lower than naive T cell. That is a well-known fact in a transplant, allo transplant setting. In terms of rejection, yes, they can reject, but as we said, these are not meant for anti-tumor effect where you don't need them to be there for long term. They are just there to bridge over the period when the patient is very immunocompromised to help to get rid of the virus. And then over time, the patient will recover their antiviral immunity and there is no need. Then the patient will reject these T cells and there's no need for these T cells to be persistent long term anyway. Yeah.
We have another question from Dan Liu. Are there any manufacturing challenges?
I think technical wise, we have yet to come to manufacturing these virus specific T cells as that I have presented. From our own experience, I cannot share much because we have only done all this work at a research scale, meaning at a small scale like a 2 cm square or 10 cm square G-Rex flask. We have optimized the method to generate in 100 G-Rex 100, that means 100 cm base area flask with not much problem, but we have not tried to validate it yet at a GMP lab. There may be some logistic issue. One of them is one of them is logistic. For example, all these studies, all this research work we have done has been using fresh donor cells. So donor came, donate platelet, we took their cone and we start the culture. For logistic, for convenience, for a third-party bank, we may not be able, because of the GMP scheduling and all these logistic considerations, we may not be able to do the culture fresh from the collected cones. So if we freeze the cone cells and thaw them and generate the cells, whether the expansion, the viability, the expansion, the response to virus peptide stimulation will be inferior. These are things that we have yet to explore. We have tried using frozen cells to generate in large scale, 2 times, and so far it has been okay. But we have yet to see with more experience.
Thank you, Prof. Lin. We have another question from Dr. Michaela Singh. Hi, Prof. Ching, enjoyed your talk. Can I ask if there are any factors you consider in selecting the multimer peptides for 5-in-1 VST? Also, whether the serotype of viruses matter.
Sorry, sorry. So is there any?
Okay, let me repeat. Can I ask, are there any factors you consider in selecting the multimer peptides for 5-in-1 VST? Also, whether the serotype of viruses matter.
Yeah, okay. So for the for the 5-in-1 VST, we just follow what people have used. The commercially available peptides are from JPT and Miltenyi of GMP grade. We use at least 2 or 3 of the pool peptides, and these are fairly standard, which is used by all the publications from those centers who have done this. So this is not any consideration, but just following what people do. Okay. The other thing about zero status. So unfortunately, clinically, we can only do CMV and EBV status. We cannot do adenovirus, HHV6 or VKV serology of the donor. So that is a problem. But it will not be a problem for our third-party bank, because in our case, we have already characterized them. We have already, without knowledge of their sero status, just generate their T cells, and we have a list of donors whom we know whether they are good killers against how many of these 5 viruses. So when we want to do the large-scale generation to really put their VST into the bank, we will definitely choose those donors who has a broad spectrum of viral reactivity based on our small scale data that we have collected, and without even knowing their sero status. But presumably, if they are good killers with a good cytotoxic T cell, they must be seropositive. Does that answer your question?
Okay, at this point in time, I don't see any further questions from the audience. So perhaps we will wrap up our session. On behalf of ACTRIS, thank you very much, Prof. Lin, for the enlightening presentation. And to all our audience, thank you for attending today's lecture.
Okay, thank you very much.
Okay, have a good afternoon, everyone. Thank you.
Thank you.